Dear students, today I am going to explain you about my second part of chapter sexual reproduction in plants and that microsporogenesis that we have already done in the first part. This is about the female reproductive unit, which is also known as pistil. Now, pistil is the female reproductive part of the flower. As we all know that this part it is actually attached to the flower with the help of thalamus. Then it's having a bulging part ovary, and over this a cylinder like structure that is style, and above it stigma are present. Now, any flower it can be mono if it is only one single pistil is present in it, bi or tri or polycarpillary. Why we are using the word carpillary? Because pistil are also known as carpels. So if there is only one carpel, single pistil is there, that is monocarpillary. If two, bicarpillary, tricarpillary or polycarpillary. Now if many carpels are present, so like in this figure you can see that these are fused together. So fused pistils are known as syncarpus, and if these pistils they are open or free, they are, for this we are using the term apocarpus. Now each pistil that I have already mentioned you is having three parts: ovary, style and stigma. Now ovary is having one or more cavities called as locules, and I can show you this in the another picture. And placenta in locules bears ovules. So number of ovules may be one, like in wheat, paddies, rice or mango there is only one ovule, but there are many ovules which are present in papaya, watermelon and orchid. So this ovule is also known as megasporangium. One ovule is known as megasporangium, and in plural it is known as megasporangia. Now from this picture it will be clear that this is actually transverse section of ovary, and here these chambers these are known as locules. So because three chambers are there, so it is trilocular. Then these chambers they are separated by these margins which are known as septa, and in the center you'll see that these ovules they are attached to the this central mass of cells known as placenta. This is through a stalk like structure. So all ovules they are attached with the placenta through stalk like structure. Next is regarding ovule or megasporangium. As we have already seen in that picture that ovule arise as primordium on placenta. You can see that ovule is actually arising as a primordium from placenta. Now the short stalk which attach ovule with placenta is known as funicle. The primordium grows into a mass of cells forming nucellus, that is the body of ovule. Again, if we'll check this body of ovule, this is actually nucellus. The two protective covering, so here you will check that this part is actually stalk, which is stalk of the ovule also known as funicle, and the point from which it is attached with the placenta that is known as hilum. And these are the two coverings of the nucellus which are known as integuments, outer integument and inner integument. Now except at the tip, this is the tip part, we are leaving a small opening known as micropyle. So this is regarding structure of ovule. So if we have to explain about ovule structure, so in this we are having funicle, point of attachment is hilum, then two integuments are there, and behind this nucellus or nucellus is present. Inside the nucellus embryo sac is there, and the opening part is known as micropylar pole or micropyle, and the end which is opposite to micropyle is known as chalazal pole. Next, regarding the structure of ovule and megasporangium, again, mainly if you have to check the basal part of the ovule is called as chalaza. This chalaza end, this end is opposite to the micropylar end. Then cells of the nucellus, inside this nucellus is present, which are rich in reserve food. So all the cells which are present in this region, nucellus region, they are actually providing nourishment to the embryo sac. So these are rich in nutrients. Next, a single embryo sac or female gametophyte is located in the nucellus, and this embryo sac is forming from the megaspore. Now it will be more clear from this diagram also. And the process of formation of this embryo sac, it is known as, or a megaspore is known as megasporogenesis. And how this megasporogenesis or development of female gametophyte occurs. Now here you will check that in the nucellus region, one of the nucellar cell is actually bigger in size, and this is also known as archisporial cell or megaspore mother cell. Now, in this cell, this one nucleus is present. It divides meiotically. So, when it will divide meiotically, meiosis 1 and meiosis 2 will occur. So, when meiosis 1 happens, then two cells will form megaspore dyad, and after meiosis 2, four cells megaspore tetrad will form. Now, out of these four cells, three cells degenerate, only one megaspore remains functional. Now, one megaspore mother cell which remains functional, this divides with first mitotic division into two nuclei. Then two more mitotic divisions takes place, like after second mitotic division 4 cells, after third mitotic division, not cells, nuclei, after third mitotic division 4 nuclei will form. Now there you will check there is no cellular division, cytokinesis is absent, only karyokinesis is going on. That means only nuclear divisions are going on in these. It will be clear from here, like one nucleus was present, first mitotic division, and then these will form 2, then 4, from 4 to 8 will form. So that means all mitotic divisions, they are free nuclear divisions. Now you can check here that 6 of 8 nuclei, they are then surrounded by cell walls and organized into cells. Now what will happen that three nuclei, they with the cell walls, they will get arranged at chalazal end, and these cells are known as antipodals. On the other hand, on opposite end at micropylar end, then again three cells are there, where one cell acts as a egg, another two as synergid. All these three, two synergids and egg, these form egg apparatus. Then in the synergids, filiform apparatus also is present. These are leaf-like structures, and these filiform apparatus, leaf-like structures, help in entrance of pollen tube during the time of fertilization. Now two polar nuclei, these are surrounded by one cell wall, and this cell is known as central cell. So if you will check in totality, there are 8 nuclei present, and 7 cells are there, 3 of the egg apparatus, 3 of antipodals, and 2 and 1 is the central cell. So we can say that embryo sac is having 8 nucleate and 7 celled stage of the plant. Now before going for fertilization, let me explain you that what is pollination, because till now in part 1 we studied about the formation of pollen grain, and in part 2 about the embryo sac. Now here pollination, as both the gametes in the plants like pollen grains and egg, these are non-motile. So they are to be brought together for fertilization to occur. to achieve this, pollination is required. Flowering plants, they have amazing methodology of adaptations to achieve pollination, and that is through external agents. Pollination is the mechanism to transfer pollen grains or shedding of pollen grains from anther to stigma of pistil. Now there are three methods of pollination: autogamy, जिसको आप self pollination कहते हैं, geitonogamy and xenogamy. Now autogamy is further of two types, chasmogamous and cleistogamous. Now first one is autogamy. Autogamy means auto. Auto means same. So when pollination is achieved in same flower of same plant, so transfer of pollen grains to stigma of same flower. There is synchrony in pollen release. So how can it be possible? जब तक दोनों में सिंक्रोनाइजेशन ना हो, पोलन ग्रेन में, पोलन रिलीज में और स्टिग्मा रिसेप्टिविटी में, so that it can accept pollen grains simultaneously. Secondly, anther and stigma should be closely placed in the flower for self pollination. जो आप यहाँ पे फिगर में देख पा रहे हो कि ये जो पार्ट है, दिस इज स्टिग्मा, दिस इज एंथर, और ये सेम फ्लावर की हैं, तो ये सेल्फ पॉलिनेशन है और ये क्लोजली प्लेस्ड है. नाउ नेक्स्ट, ऑटोगैमी के अंदर टू टाइप्स ऑफ फ्लावर्स है. इन केस ऑफ यू कैन चेक दैट flowers which are similar to the flowers of other species with exposed anther and stigma, that are chasmogamous. Similar flowers हैं, similar species की हैं. Then flowers which do not open at all, that is cleistogamous, जिसमें self pollination होने के ज़्यादा chances हैं. अगर हम देखेंगे, plants like viola, oxalis and commelina produce two types of flowers, chasmogamous and cleistogamous. एक ही plant में दोनों तरह के flowers हैं. Cleistogamous flowers are advantageous because there is assured seed set even in the absence of pollinator. आपको इस पिक्चर से ये और क्लियर हो जाएगा. देखो, ये जो फ्लावर है, इसमें ये चेस्मोगैमस फ्लावर है, ये ओपन होगा, और ये इसके नीचे क्लिस्टोगैमस फ्लावर है जो ओपन नहीं होता. सो चेस्मोगैमस जो होता है, विच आर similar to flowers of other species with exposed anthers and stigma. So there are chances of cross pollination in this. और self pollinated तो है ही क्योंकि same plant है. Now cleistogamous which do not open, in such cases anther and stigma, they lie close to each other, and when the anther dehisces in the flower buds, pollen grains come in contact with the stigma to effect the pollination. Next comes geitonogamy. Geitonogamy, where transfer of pollen grain from the anther to the stigma of another flower of same plant. Means एक ही plant है, उसमें दो flower हैं, एक flower का pollen grain दूसरे flower के stigma पे जब fall करता है, पर वो move कैसे करेगा? Through pollinating agents, which may be wind, water और insects etc. और birds also. So genetically अगर आप देखोगे कि these are similar to autogamy because pollen grain is of same plant but flower different है. Example is corn. Now next comes after geitonogamy, then next is xenogamy. So xenogamy is cross pollination, is also known as allogamy. So cross pollination is the transfer of pollen grains from anther of one flower to the stigma of a genetically different flower. So it is performed with the help of external agency, again abiotic or biotic कोई भी हो सकता है, through wind, water or insects. Example is onion and broccoli. So in this part, in the second part, I told you about megasporogenesis and how pollination can occur by three methods, that is allogamy, autogamy and geitonogamy. Then after this we'll study about double fertilization, apomixis and polyembryony in the last part of this chapter. Thank you so much.